Now, very often, when an antigen first comes into the body, causing an infection, the first cells that recognise the presence of this antigen are called antigen-presenting cells, or APCs. And antigen-presenting cells, or APCs, are located where you would expect them to be guards of the body. So you get them, for example, in the skin, just in the lower layers of the epidermis and in the dermis. And you get them in the mucous membranes. So they're guarding the body against potential infections. And let's look at a couple of examples of antigen-presenting cells now. Well, one is the monocytes that we're already familiar with. What do we call monocytes when they're present in the tissues? When they're in the tissues, we call them macrophages. So macrophages can be the first cells that detect the presence of an approaching antigen. And there's another type of cell that's like spread out in the tissues, so it can detect anything that's floating past. It's a bit like an octopus, really. It's got arms. And it's called a dendritic cell. Dendrites are these long arm-like extensions. It's got a nucleus in the middle. And again, if any antigens are in the area, it can be detected and picked up by this cell. Now, in the case of the macrophages, as you know, the antigen is actually ingested and digested in the process of phagocytosis. But what actually happens as well is some of this antigen is processed and broken down, and it's actually presented on the surface of the macrophage. And it's not presented as it is, it's presented in combination with some proteins from within inside the cell. And these proteins, the body proteins that are presented with the antigen, are called the major histocompatibility complex proteins, the MHC proteins. So what actually happens is, there's a couple of MHC proteins here. These derive from the cell itself, and these present the antigen to the outside world, that fragment of antigen. So it's presented. So what the macrophage is presenting is some MHC proteins, major histocompatibility complex proteins, and a fragment of the antigen. And much the same happens with the dendritic cell. Again, this presents it. On the surface membrane, it's combined with MHC proteins. And again, this presents fragments of the antigen. So the antigen-presenting cells are guarding surface areas of the body where infection is likely to get in, where the infection is likely to creep in. When it comes in, they're detecting its presence, they're capturing the antigen, they're processing it, and they're presenting it on their cell surfaces in combination with MHC proteins. Well, what's the purpose of all this? Well, we said that the B cells will react with a particular antigen. So the B cells, there's a particular antigen, the B cells can recognise that, and when that activates the B cell, there'll be clonal expansion in the B cell. But the B cell actually will respond to it much better if it's attached to a major histocompatibility complex protein. So there's the antibody on the surface of the B cell. There's the B cell. It will respond to just the antigen itself, but most B cells, or most antigens, are much more stimulating to the B cell if they're in combination with this major histocompatibility complex protein. So the antigen is processed by the antigen presenting cell and then presented on the surface. It's a bit like saying, well, here it is, do something with it. And what the B cell does is the combination very quickly activates the B cell to undergo the process of clonal expansion. But with T cells, antigen presentation is absolutely essential. The T cells, remember the T helpers, the T suppressors, the T cytotoxic cells, they will only be activated to undergo clonal expansion if they are first activated by a combination of the MHC protein and the antigen, in this case presented by the dendritic cell. So what you have on the surface of T cells, they don't have antibodies on their surface. Only the B cells have that. But on the surface of all the T cells, there are molecules, protein molecules, which will recognise the presence of antigen. And particularly, well, exclusively, the receptors on the surface of T cells. Here's the T cell here. The T cell will only recognise antigen in combination with major histocompatibility complex proteins on the surface of an antigen presenting cell, an APC. So the T cell can be activated in response to a combination of MHC and antigen on the surface of a dendritic cell, or indeed another APC such as a macrophage. And then again, once these cells have been activated, this will undergo clonal expansion, giving rise to a population of B cells. The effector B cells are called plasma cells that produce the antibodies. Likewise, in the same way, once the T cell is stimulated by the presence of the antigen and the MHC protein as presented on an antigen presenting cell, the T cell will undergo a process of clonal expansion as well. This means we have lots of T helper cells. This is a T helper cell that will stimulate the B cells. Or if it's a T cytotoxic cell, again we'll get a clonal expansion of those, and we'll see what they do in the next section of this talk. So antigen presenting cells guarding the surface of the body, taking in antigen, processing it, presenting it to the lymphocytes. The B lymphocytes responding better to presented antigen, the T cells responding exclusively to presented antigen, to bring about the process of clonal expansion, the effector cells, which get rid of the specific infection. So the APCs are sometimes considered to be a link between innate immunity and specific immunity. The antigen presenting cells will recognise and process any antigen, they're non-specific, but they will be, by presenting the specific antigen with the MHC, they will stimulate the specific lymphocyte-mediated acquired immune response.